So a big thank you to the organizers of this COVID-19 virtual session for giving me an opportunity to speak about the biology of SARS-CoV-2. It's really and indeed a pleasure and an honor for me to be interacting and sharing some of my what I have learned over these past few months about the biology of SARS-CoV-2 and how it is different from the previously known SARS and the MERS viruses. So I am Dr. Aparna Mukhopadhyay from the Department of Life Sciences, Presidency University, Kolkata, India. So first a little bit about coronaviruses. So these are actually enveloped viruses, meaning that they actually steal an envelope from the host in which it evolved. So from this envelope emerges the spike proteins, which are the surface glycoproteins of the virus itself. The genome is a plus-stranded RNA genome, which means that it is capable of synthesizing proteins as soon as it enters the cytoplasm of the host. These particular viruses are about 160 nanometer in size. They actually have one of the largest genomes for being an RNA virus, and they are actually zoonotic viruses, and I'll be telling you a little bit about them very soon. And this particular coronavirus, the SARS-CoV-2, is actually the 7th member of the human coronaviruses that we have known so far. So coronaviruses actually get their name from the crown-like appearances you get to see when you look at these viruses under the electron microscope. And it actually comes from the surface proteins, which are known as the spike proteins. So they belong to the order of nidovirales, and they have 4 genera, alpha, beta, gamma, and delta, of which alpha and beta genera viruses infect mammals, and gamma and delta infect primarily birds. Now, among the beta genera, there are A, B, and C lineages, and some of the well-known viruses are listed here. So these are some of the major viral outbreaks, of which, like I said before, this particular coronavirus, the SARS-CoV-2, is the 7th coronavirus that is known to infect humans. So we have been studying them from the 1960s, but it actually has been known for a very long time. So these are 4 other coronaviruses which are known to infect humans, but they are not very pathogenic. Since the beginning of the century, coronaviruses have brought themselves into fame, or rather defame, when there was the first outbreak of the SARS coronavirus in 2003. And then in 2012, there was an outbreak of the Middle Eastern respiratory virus, which is also known as MERS. And then in 2019 came the SARS coronavirus 2. So I will be distinguishing this virus from the previously known SARS coronavirus, as this is known as the SARS-CoV-2, and the previously known SARS coronavirus is known as SARS-CoV. If we were to look at the pathogenicity and look at the fatality rate, this particular coronavirus, the causative agent for COVID-19, happens to have the lowest fatality rates compared to the previously known SARS and the MERS virus. But in general, they have a pretty high fatality rate when we compare them to seasonal flu. So talking about the zoonotic origin, all of these viruses actually evolved from bats. But from the bats, these viruses had an intermediate host, which happens to be civets for the first known coronavirus, camel for the MERS, and pangolin for the SARS-CoV-2. So if we were to look at genomic similarity, there is 89% similarity of the SARS-CoV-2 with the bat-derived coronavirus, and 79% similarity with the SARS-CoV, and about 50% similarity with the MERS. But if we were to go in a little further and look at the receptor binding domain of the S protein, we see that it's actually similar to that which is present in pangolin. So this is what happens is that the bat coronavirus happens to find itself in the intermediary host, where it seems to take up some of the properties of the virus which is present in the intermediary host, and then lands up in the human, where it again goes through certain amount of adjustments. Now, where all these live animals are actually stored in one location, which was one of the live markets in Wuhan, China, these things are possible to happen, and that is why there is a big possibility that the virus actually emerged from the live markets, live animal markets of Wuhan, China, where the virus emerged, which originated in bat, passed through the pangolin, and finally landed up in the human and became a pretty pathogenic virus. So, like I said before, at the genomic level, the SARS-CoV-2 shares about 79% similarity with SARS and about 50% similarity with MERS. But if we look at the spike protein, the spike protein of SARS-CoV-2 has 75% similarity with the SARS-CoV. But if you look in a little further and look at a particular subunit of SARS protein, there is about 65% amino acid identity. So this is a genomic organization and the proteins that are encoded by it. So these RNA viruses are actually single-stranded RNA genome, which has a 5' cap, and it is a positive stranded RNA molecule, which ranges from 26 to 32 kilobases in different types of coronaviruses. So the RNA includes at least 6 open reading frames, or ORFs. The first ORF, the ORF 1A, 1B, comprises about 2/3 of the genome, and it encodes the replicase proteins and some of the important proteases about which I will be telling you soon. Then comes a large number of open subgenomic, excuse me. So the genome encodes for these. So from the, what happens is from the 2 overlapping ORFs, 1A and 1B, 2 large polyproteins, PP1A and PP1B are translated. And the rest of the genome is transcribed into a nested set of subgenomic mRNAs. From these comes all the structural proteins and few of the non-structural proteins. So what is important is that the first part of the genome which is translated comes the proteases that are important and the RNA-dependent RNA polymerase. So this is the genomic structure once again, and what is present in the SARS coronavirus is there are 4 structural proteins. First of them is the spike protein, which happens to be the most important when we talk about antigenicity and viral entry, because this protein is responsible for entry of the virus into the cell. Then there is another membrane protein, which is known as the M protein, which is responsible for the internal viral cell shape and also triggers assembly. The nucleocapsid protein, or the N protein, binds the viral RNA and elicits a cell-mediated immunity. And the envelope E protein also helps in the assembly and release of the virus particles and also acts as an ion channel. Apart from that, there are 16 non-structural proteins. And of these non-structural proteins, NSP3 and NSP5 encode for 2 very important proteases which help to generate the mature protein which is responsible for the virus replication for the entire life cycle of the cell. The RNA-dependent RNA polymerase, or the polymerase which is involved in the genome replication, is encoded by NPS NSP12. Then there NSP8 encodes for a primase. NSP7, 8, 9, and 10 are all involved in some steps of the replication process. NSP14 is a 3' to 5' exonuclease, which is one of the unique features of coronaviruses, where all the errors made by the RNA-dependent RNA polymerase get actually corrected by this exonuclease, and therefore helps in maintaining the fidelity of the RNA transcription. NSP16 is a S-adenosyl I-1 methionine dependent RNA transferase, and it is also involved in the cap formation. So if we were to move on further, so these are comparison of the genome structure between the SARS-CoV-2, the SARS-CoV and the MERS coronavirus. And as you can see that although they are very similar, there are actually certain differences. The differences lie in the spike protein, in the transcript, in the reading frame 3, as well as in the reading frame 8. These are actually the major differences between the SARS-CoV, SARS-CoV-2 and the MERS coronavirus. So looking into a little more detail of the spike protein, this is the major structural glycoprotein which is present on the surface of the virus, which is responsible for entry. So the spike protein exists as a trimer, but it is subdivided into two units, the S1 subunit and the S2 subunit. The S1 subunit has the receptor binding domain by which it is capable of interacting with the ACE2 receptor, which is the most well-known receptor for the SARS-CoV-2. The S2 subunit bears the fusion peptide by which the virus is able to allow the fusion of the host and the viral membranes. This is a cryo-EM structure of the spike protein from SARS-CoV-2, and what you can see highlighted here is all the different glycosylation and other motifs which are unique to either the SARS-CoV-2, or they were exclusively present in either the pangolin, or they were common to both the bat as well as the pangolin. So what is important here is there is a furin cleavage site in the SARS-CoV-2 S protein structure, which was not present in either the pangolin or the bat, and was not present in the previously known SARS-CoV either. And I will be coming to that a little while later. So if we were to compare the biological features, I have already talked about the homology. They all originated from the bat, but then passed through different intermediate hosts. And this is the different lineages of the beta coronaviruses. The receptor that is being used is ACE2 for the SARS-CoV-2 as well as for SARS-CoV, but MERS coronavirus uses a different receptor which is known as DPP4. Apart from that, the SARS-CoV-2 has a multi-basic furin cleavage site, which was not present in the previously known SARS coronavirus, but is also present in the MERS coronavirus. So this is exactly how viral entry happens. So this is the receptor ACE2 present in the host cell, and this is the spike protein which is jutting out from the surface of the virus particle. So the receptor binding domain of S1 binds to the ACE2 region, ACE2 protein present on the host cell. After interaction of the RBD domain with ACE2, there is cleavage of the junction between S1 and S2, and this is mediated by two different proteases about which I will be telling you very soon. So as soon as S1 and S2 junction is cleaved, the fusion peptide, which is located within the S2 protein, is exposed, which will now insert itself within the host protein, the host cell membrane, and thereby bring these two membranes close to each other so that they may fuse and allow the virus to enter the cell. So this is something about the biodistribution and the host cell receptors of SARS-CoV, MERS, and SARS-CoV-2. So what you can see here is the SARS-CoV enters through ACE2. SARS-CoV-2 also enters by ACE2, but MERS enters through a different receptor which is known as DPP4. So ACE2 has a very vast distribution, which includes the respiratory tract, the GI tract, heart, kidney, olfactory neuroepithelium, and few other organs. DPP4 is also expressed in the liver, and a few other cellular, which gives pretty cellular and tissuism is viral. Now we know well aware that the lungs are the main target organs of SARS-CoV-2 because it is coming in through the airways, and that is how this is the first layer of cells that the virus is being exposed to. So the ACE2 is actually expressed in all these cells, particularly in the human lung cells. Majority of them are type 2 alveolar cells. Other lung cells are the type 1 alveolar cells, the endothelial cells, and other airway epithelial cells. So apart from that, we are also aware that this virus is capable of causing GI problems as well as cardiovascular problems and kidney problems. And all of them is particularly because there is an abundant expression of ACE2 present in all of these organs, which is why if the virus is out in the systemic circulation, each of these systems can get affected. And that has been seen in many of the serious cases of COVID-19 patients. So this is an alignment of SARS-CoV receptor binding domain with that of SARS-CoV-2. What you can see in white is the receptor binding domain of SARS-CoV, and in green is the receptor binding domain of the SARS coronavirus 2. And what you can see is that although these two regions are very similar to each other, but there are subtle differences between the two. And so if the sequences of each of these virus, which is SARS-CoV-2, pangolin SARS-CoV, bat coronavirus, and SARS-CoV were all to be aligned, you can see that although they are very, very similar, there are certain differences between each of them in the sequence level, which is reflected in the structural level, which is also reflected in the functional level. And that is why there is differences between each of these virus in terms of the pathogenicity as well as in the lethality that we are getting to see. So is there any difference between receptor binding capabilities of the SARS-CoV versus the SARS-CoV-2 with respect to ACE2 receptor? There are certain kind of conflicting reports. One report was there about an 10 to 20-fold higher affinity for the SARS-CoV-2 spike protein for the angiotensin converting enzyme when compared to the SARS-CoV. There are other reports that talk about the receptor binding domain of SARS-CoV-2 having been lies in a lying down position, which results in an ineffective receptor binding, and it also helps in immune evasion. Whereas the receptor binding domain of SARS-CoV happens to be in a standing up position, which helps in a highly effective receptor binding, but also helps in immune recognition. So this is an area which needs to be further investigated to find out whether there is actually a better or a less interaction of the spike of SARS-CoV-2 with ACE2 as that of SARS-CoV. When we look at specificity, it has been known that SARS-CoV exclusively binds to ACE2 and the MERS coronavirus exclusively binds to DPP4. The SARS-CoV-2, what we have seen in maximum literature, talks about ACE2 as the most preferred receptor. But there are a few reports that also talk about DPP4 as also an alternative receptor that allows the virus to enter the cell. So this is again something which needs to be investigated further to find out how much is the receptor specificity for this kind of virus. So the next question is that are the epitopes of the spike protein of SARS-CoV anyway similar to that of SARS-CoV-2? So is it possible that any patient serum that had been obtained from people infected with the previously known SARS-CoV, do they have any cross-reactivity with the people infected with the SARS-CoV-2? So this is an experiment where they have taken antibody which was generated against the spike protein of SARS-CoV and looked at cross-reactivity. So there is cross-reactivity with that of SARS-CoV, but there is no cross-reactivity with that of SARS-CoV-2. And if the receptor binding domains were swapped, then there is loss of cross-reactivity. On the other hand, here is another experiment where people have taken COVID-19 patient serum and looked at whether it is possible to inhibit viral entry. And they see that COVID-19 patient serum inhibits the entry of SARS-CoV-2 pseudoparticles, but has no effect on the entry of pseudoparticles from SARS-CoV. which indicates that the epitopes of the two viruses are different, which is why antibody of one does not cross react with the other protein. So a little more, if we were to look at, I have already spoken about the first 2 points, but if we were to look at a little more detail, there are distinct epitope features between the RBD of SARS and the C-terminal domain of SARS-CoV-2. Although they are structurally very similar, but the epitopes happen to be different. Again, the N-terminal domain of MERS coronavirus prefers alpha-2,3 linked sialic acid over an alpha-2,6 linked sialic acid. But how much this is biologically important, that still needs to be investigated. So I did mention that there is a furin cleavage site. So this is the spike protein of SARS coronavirus, and this is the spike protein of the SARS coronavirus 2. And what is seen is the SARS coronavirus 2 has a multi-basic furin cleavage site, which was absent in the previously known SARS coronavirus. Now, what is the importance of this furin? We have to understand that lungs express huge amounts of furin, and it is also present in the respiratory tract. It has been shown that both furin as well as another protease, TMPRSS2, are important in the cleavage of S1-S2 interface. And this is one of the important steps, as I showed you before, for the virus to enter the cell, because this is one of the important steps that allows the viral and the host membranes to fuse. And it has been seen that if any of these sites were disrupted, that is disruption of the furin or the disruption of the TMPRSS2 sites, results in loss of viral and cellular and viral membrane fusion, which means viral entry is inhibited. So both of these enzymes are actually helping the virus to enter the cell. Furthermore, other publications have shown that the furin also helps, the furin cleavage site also helps in cell-cell fusion and helping the infection to spread from one cell to the other cell. So these are cells where there has been a spread of infection, whereas in these cells, the infection didn't spread. So what is important is the furin cleavage site, which is present in the SARS-CoV-2, is actually helping the virus to spread infection, first and foremost, get infected, and secondly, spread the infection from one cell to the next. So this is proposed life cycle of the SARS coronavirus, which is not completely worked out now, but it is kind of proposed based on what we already know about the biology of coronaviruses and the recent flood of literature that has generated in these past couple of months. So the virus enters the cell by binding to the receptor ACE2, and then it comes in by a method called endocytosis. During this process of endocytosis, there is fusion of the viral and the host membranes, by which the genome is released into the cytoplasm of the cell. Now, as soon as the genome is released, as I had showed you earlier, the two polyproteins are generated, PP1A and PP1AB, that leads to the synthesis, which generates these important proteins which are involved in the RNA replication process. Apart from that, the subgenomic minor strand RNAs are also generated, from which the viral structural proteins are synthesized. All of these, all the genome as well as the structural proteins come together, and the ER-Golgi intermediate compartments are the sites of assembly, where the final virus particle finds itself in a secretory vesicle and can ultimately leave the cell via the secretory pathway. Excuse me. So this is a summary of the life cycle of SARS coronavirus with MERS coronavirus. And as you can see that although in general, they all look kind of similar, but if you were to look at the nitty-gritties of the biological details, they would differ. This is a little bit about the replication process. So the positive stranded RNA genome is translated to generate the replicase proteins from the open reading frame 1A, 1B. These proteins use the genome as template to generate the full-length negative sense RNA, which subsequently serve as templates for generating additional full-length genome. So the coronavirus mRNAs all contain a common 5' leader sequence, which is fused to downstream gene sequences. These leaders are added by discontinuous synthesis of minor sense subgenomic RNA using the genomic RNA as a template. Subgenomic RNA synthesis RNAs are initiated at the 3' end of the genome, and they proceed until they hit one of the transcriptional regulatory sequences that reside upstream of most open reading frames. Then, through base pairing interactions, the nascent transcript is transferred to the complementary leader TRS, and then the transcription continues through the 5' end of the genome. So these subgenomic RNAs serve as templates for viral mRNA production. The assembly of SARS coronavirus as well as SARS coronavirus 2 is been seen to occur in a reticular-vesicular formation within the endoplasmic reticulum. So strange things have been seen where they see the formation of these double membrane vesicles present in this reticular-vesicular network, which is part of the ER and the Golgi apparatus. So over here, people have seen the presence of the non-structural proteins. So it is believed that in this network of membranes within these double membrane vesicles, this is where the entire replication of the process of the virus as well as the assembly of the virus happens. So this is just a very small snapshot of the immune response that happens. So it is well established that due to the viral infection, there is going to be an interferon response because of the presence of the viral genome within the cell. It is been shown very well that both SARS-CoV as well as MERS have different ways by which they are able to inhibit the type 1 interferon system and within the cell. So much of this is still unknown exactly what is happening in the SARS-CoV-2. A little bit of it has emerged, but it is believed that much of the system where there is inefficient activation of the type 1 interferon system happens with SARS-CoV-2, which is probably somewhat similar to what has been happening for SARS coronavirus as well as in MERS. And this is one of the reasons why there is the generation of the cytokine storm. So concluding remarks and the take-home message is that coronaviruses have been neglected over time because the SARS coronavirus fizzled out and the MERS didn't really spread. And then came 2020 when scientists all over the world were forced to drop everything else and start thinking about the importance of coronavirus. So in spite of the huge volume of research on SARS-CoV-2, there is much of the biology that still remains an enigma. But there are many unique and interesting features to tap and explore, and unless we really tap and explore and really understand the virus, we will not be able to predict exactly how to develop a therapeutic or even predict what will be the ultimate fate of COVID-19. So as an acknowledgement, these are all my lab members who help out with all the workings in the lab, and thank you